Okay, so today we're talking about motor learning and also the basal ganglia. Okay, now let's talk about motor learning first. And now when it comes to learning, the first thing you think about is how the synapses are being modified. And you'll see that when it comes to motor learning, a lot of the data, especially when it comes to animal researchers, they've been doing a lot of work in the primary motor cortex. So the motor cortex really, so the first thing you'll see is that motor learning induces a rapid formation of new dendritic spines or synapses that are basically maintained over time and they are also functionally clustered. And then erasing these new spine formation can also erase motor learning abilities. And then the last part about motor learning we're going to talk about today is that knocking out specific genes can really alter like the learning basically. And then now it really doesn't matter if you knock them out globally or you just kind of knock them out in specific cells such as the glial cells. And all of these can impact the motor learning effect. So the first thing that people wanted to do is they want to make sure that animals can really learn. So in rodent models, what they have to do is they want to make the animals want to perform a certain task. So in this study, what they did is they have a mouse put in like a specially made chamber, and then they will food deprive the mice. So this will provide the motivation for the animals want to use their forepaw to grab food. So they will place a food reward here, and what the animals have to do is they have to use their paws to really reach for the food pellet. And over here in B, where you can see that there are animals that are really learn this pretty fast because they don't want to starve to death, right? And then there are some animals that just like they just no matter what they cannot learn the task very well. Now, out of the animals that learned really well, they want to make sure that they are imaging these are indeed in the correct areas. So what they did is they looked at the frontal lobe of these animals. And then what they'll have to do is they want to make sure that the image there where they are imaging is it's basically the correct region. So what they did is they stuck a electrode into wherever they're imaging, and then they will stimulate this area. And then what they saw is that they saw a movement of the forepaw, and therefore they are imaging where the forepaw, that the forepaw is happening. Right? And then what they did is they will look at the dendritic spines here and also the dendrites, and then they will look at in the control animals where they were not learning at all, and then they'll look at the ones that were trained, and you can see that there are, and so then what they will do is they will look at the formation rate of the spines as well as the elimination rate of the spines. Okay, and then so what they saw is that even just after 1 day of training, what they saw is that the formation rate of the spines in the learners really increased, while it did not really impact the elimination rate. And then what they saw also is that the formation rate correlated with the successful reaches positively. So which means that the more new spines that are grew out, the better the animals are improving, basically. Okay, so the next thing what they wanted to do is they wanted to kind of make sure that the enhancement of the spines during the motor task is task and time specific. Okay, so in A, they're just looking at many different kind of tasks that are involved. And you can see that, especially in day 4, when you look at control, when you look at the training, when you look at the part, so they saw that there are a lot of new spines that are being generated in all of the training conditions that you see in black, but then not in any other control conditions, such as when they were looking at the training but in the ipsilateral motor cortex, they don't see any effect. When they look at the training in the contralateral somatosensory cortex, they did not see any increased spine formations. And then that when the animals that fail to learn in the contralateral motor cortex also did not see any kind of differences. So what you are seeing is that this area of enhanced spine dynamics during motor learning is task and time specific. Okay, and then now as opposed to what you saw in the previous slides, following the second day, and also the fourth day, and also the eighth day and the 16th day, what they're really starting to see is that there is a increase of dendritic spine elimination as well. So what this means is that motor learning really induced a initial additional insertion of dendritic spines, which means that there are more synapses that are being inserted into the circuit and followed by an elimination point of the dendritic spines, which means that there is an initial recruitment of additional synapses followed by additional refinement of the synapses, and this is how the motor trajectory is refined over time. And in C, what you're seeing over here is that basically this is looking at the total number of spines. So in the first day, what you see is in the red, what you're seeing is that there is an initial increase of dendritic spines, and then followed by the downfall of that, right? As opposed in the blue, what you're seeing in control animals, it's just that the number of spines goes gradually down. Okay. So what D is showing you, and E and F just kind of basically showing you the example of the dendritic spines. Okay. So the next thing what they wanted to do is to look at the fate of these new spines and how they differ in the animals that went through motor learning and the ones that did not go through motor learning. So they categorized the dendritic spines into two main classes. They will have the pre-existing spines that you see here, and then they have the new spines that you see in here. So so basically you can see that in here. So the pre-existing spines are the ones that existed in 4 days in between the first imaging and the second imaging. And then the new spines that are the ones that did not exist in the first imaging time and then but appeared on the second imaging time. And then they wanted to look at these fate of these spines. And then here just some here in B and C, just some sort of representative images that they're looking at. And now D, what they're showing you is that you can see that the dendritic spines are pre-existing, are pretty stable. They do not get lost a lot. But the spines that are new really get lost quite a bit. Okay, you can see that it went from 100% in the training session all the way down to around 60%, and then around like 50% when it comes to the control animals. But you can see that the additional training of that stabilizes the loss rate. And you can see that when it comes to the motor learning conditions compared to the control conditions, you can see that the spines that are survived with continuous training is basically stays much better compared to the new spines that just pretty much grew out in the control conditions. What this means is that in repetitive motor training um, really kind of stabilize all of these new dendritic spines that forms a long and enduring lasting motor memory. Now, the next thing what they wanted to do is they wanted to make sure that, well, where do these new spines are appearing? Okay, so what they did is, when you look at here, you'll see that in day zero, they don't see that you don't have that many spines, but then there is one that grew out here in day one, and then there's another new one that grew out here in day four. So they thought of this idea that maybe these new dendritic spines that are basically that are grew out from because of the new motor skills are because that they are functionally clustered. So then the next thing what they did is they look at the cluster analysis in the control animals versus the training animals, and then they saw that a lot of these clusterings are happening in the early stages and not so much in the late phases. Okay, and then the next thing what they wanted to do is and this is basically looking at clustered versus non-clustered in the day zero for 16 time of imaging, and then versus day zero for an adult images. What they saw is that it really doesn't matter what you're how you're looking at it, but what they're saying that there is a significant amount of the dendritic spines that are clustered following repetitive training. Now, what you're here, what you're basically seeing here is so they will it's a simulation data, and then they basically just kind of seeing that what are the amount of the cluster spines, and you can see that the persistent cluster spines are basically only takes about somewhere around 15% of all the dendritic spines, and then when you look at the kind of so all these number of the new cluster spines, what you're seeing is that the normalized spine head size grew bigger from day one to day four. So what this means is that there is a functional gain of this increased amount of plasticity that is associated with the cluster spines, and but they don't see this in the non-clustered new spines. Okay. Now, the next thing what they wanted to do is they wanted to make sure that the clustered new spines form over multiple training sessions of the same but not different motor tasks. So what this means is that they have several conditions. They will have the animals that produce exactly the same tasks over 4 days, and they'll have the cross training. So they have the animals perform one task on one day, but then switch them to another task on days 2, 3, 4, and then they have another uh, uh, another condition where they just put them into motor enrichment where the animals are running around but they're not doing any kind of training, and then they will image in day 0, 1, and 4. Okay, and then and this and B, what they're showing you is this basically all the dendritic spines activities when it comes to the reaching only condition. You can see that there were no spines here in day zero, and not so much in day one either. But then at day four, there are two spines that just pretty much simultaneously popped out. And then in CDE, what they're basically showing you is just that the spine formation rate is pretty much similar when you look at all of these different three conditions. All three conditions boost up new dendritic spines, but then the kind of dendritic spines that basically that are clustered, the only kind of part that you really see is in the reach only conditions. You don't see this in the cross training. You don't see this in the molar rich environment, which all boost up the dendritic spine formation rate that you see in D. So what this means is that the cluster new spines formed over multiple sessions really has to require the same with the same motor task, and if you kind of switch over the different motor tasks, a different set of dendritic spines are going to grow out basically. So, and this also kind of tells you that practice makes perfect, because additional practice is going to recruit additional spines and also the insertion onto the circuit. So this is how exactly basically how you get better at specific tasks. Okay. Now you're probably thinking that, well, so it seems like new dendritic spines growth is correlated with enhanced motor learning, right? So does that mean that if I can somehow make the dendritic spine not grow out, or if I can make the recently dendritic spine that grew out, if I can let them go away somehow, then does this erase the memory of the motor skill that is learned? And it turns out that yes, that is exactly the case. Okay, so in this study, what they did is they have this thing. Basically, they've called it this photo activation of RAC. This is a small protein that resides only in dendritic spines. And then what they can do is they can do photo activation. That's what PA stands for, photo activation of something. And then what they found is that before, you can see that in this AS, you can see that this basically picks out all of these new dendritic spines. But after they give the photoactivation, you can see that all of these dendritic spines that are just recently grew out gets erased. Okay, and then down here they do a lot of just a lot of control experiments to basically show you that after they use a photoactivable RAC, photoactivation of the RAC protein, they can make the new dendritic spine shrink. Okay, so that's basically just the gist. They use a lot of complicated technique, which I don't want to go over here because this is more suited for a graduate student, a graduate class. Okay, so the next thing what they wanted to do is they wanted to make sure that uh, how does photoactivation induce dendritic spine? How can they use this to erase motor memory? And so what they did is they will have the animals do certain motor training, okay, and then after they're done with the training, then they will use a photoactivable rack to basically erase these dendritic spines. Okay, and then they do this in different time points. Some of them they do this between day 2 and day 3. In this case, they did it between day 3 and day 4, and here they did it between day 4 and day 5. Just kind of different timing of that. Okay, and over here in protocol 1, what you can see here is that um, basically the amount of the so basically what you can see that the animals the after they get zapped with the photo activation, what they can do is that the animals after photo activation, they don't do as well as without photo activation. Okay, and then in protocol 2, we saw exactly the same thing. We saw that, after the photo activation, there is a significant reduction of the motor learning ability. So in this, and they did something called the roller rod, in which they have the animals placed on a balance beam, and then because the animals are afraid of falling, they need to adapt to this rotating roller rod so they don't fall on them. Okay, and now in day 3, you can see that photoactivation no longer has an effect, and that's because the dendritic spines by those days has already established, and no matter how much you try to make the existing spine that has already incorporated very much into the protocol, into the existing circuit, what you cannot do is you cannot erase a memory that is really well formed already. So they found that the best time to erase a memory is really while the memory is still being formed, but not after it has been formed. And so that's what basically this is basically what they're telling you here in figure E. Okay, you can see that in protocol 1 and 2, they were able to erase a lot of them a lot of the memories. But then in the protocol 3, and this is when they're trying to do this between day 4 and day 5, they can no longer erase the memory. So which means that there is an optimal time that you can actually erase a memory, and but not after a memory has already been formed. So this is a very critical work that basically show us that there is a very good time for us to erase a memory if you want to unlearn something. But then after you learn them and you have already been solidified, you cannot unlearn them because they are already embedded in within your motor circuitry. Now, in this last slide, I want to talk about Fragile X. And so the Fragile X is basically a disease that people really have a hard time learning. And then this is categorized by, well, something like a very low intelligence level. So this is why they call the Fragile X mental retardation protein, which means that if you knock out of this protein, which a lot of Fragile X patients have, then in the animals you can reproduce an inability to learn in in mice. Okay, so basically, so when you knock out the FMR1 gene, which is responsible for making the Fragile X mental retardation protein, then what happens is that the animals don't learn very well. Okay, now let's take a look at this group. What they did is basically they trying to see whether or not the Fragile X mental retardation protein is it more important in neurons or is it more important in glial cells? Now, and they're looking at a very specific kind of glial cells, which is the astrocytes. Now, one of the things that you might not know is that the Fragile X mental retardation proteins are really residing not only in neurons but also in astrocytes, and they also reside in other glial cells such as the microglia. And so what happens when you look at so what they're trying to do is that they wanted to kind of make sure that they are really identifying the glial cells, and this is what they did with a very specific glial line, and so they picked it with something called the TD tomato, which is a reporter line, that, and when they cross them with a very specific Cree line, and then what they can do is they can show that these are exactly the kind of cells that they are targeting. So whatever whenever you see a TD tomato positive cells, then that basically is showing that this is what the Cree line is working on. And then what happens that what they're looking at is they want to make sure that what are these exactly TD tomato positive cells that is shown in red, and then so when they looked at them, they stained it against S100, which marks the glia cells. They can see a perfect co-labeling with that, but they don't see this with the new N, which labels neurons. They don't see this co-labeling with the allyl 2, which labels the oligodendrocytes, and they do not see this co-label with the IBA1, which labels the microglia, which means that this very specific line is targeting uh, the astrocytes and not anything else. So they show this in vivo here, and then down over here on the right, what they're just kind of showing you is exactly the same thing, but in culture. So this is in culture what so they would use so they're basically looking at the S100 staining with the phalloidin, and then you can see that this is a very good co-labeling here. Okay. Okay, so next thing what they wanted to show is that whether or not knocking out the FMR gene, the Fragile X mental retardation gene in globally or in astrocytes specifically can actually perturb motor learning, and it turns out that it does. And so over here you can see that in black this is the wild type animal, and then the in blue is what I'll call like the fake knockouts, because this is a a this is basically a flux animal without the Cree, so without the proper combination. All right, and then you can see that both of these in black and in blue, they perform pretty well by the last 3 days. Now, when you have the FMR gene knocked out in the astrocytes, which is what you see in red, you can see that the animals do not perform as well as the controls. So you can see the success rate is much lower than the control animals. And then the animals that is a global knockout that has the FMR gene knocked out in both um, neurons and all the glia cells, you can see that they really do the worst. This is what you see in the green trace. Okay, and now in B, what they're showing you is that, do you remember that in the previous slides that dendritic spines grew out as a result of motor learning? So they also wanted to make sure that whether or not this is true. And then so they were looking at the dendritic formation rate that are basically due to the improvement of the reaching task. You can see that so the spine formation rate is not that much in all of these conditions, but so basically what they're showing you here is that the knockout they so the knockout animals, it doesn't really matter if they are knocked out in the astrocytes or globally knocked out, they do not have a new increased dendritic spine formation, okay, or elimination for that matter. Okay, so which means that there is no learning and there is no correlated amount of the that is being induced by the learning. Okay. Okay, so that was it for the motor learning. So make sure you know that that's probably going to be on the exam. Okay, and now let's talk about the cerebellum and the basal ganglia. Okay, now the cerebellum is what you see that there are three divisions of the cerebellum. So these regions differs basically in terms of their anatomical projections to the deep cerebellar nuclei and also the extracellular target regions. And so, for instance, well, you can see that this spinal cerebellum really has a lot of descending systems. They descend this to for like motor execution. But the neocerebellum, there are several part of that. So the ones I kind of really want to talk about that is the deep cerebellar nuclei. So these are going to really give rise to things like they're going to synapse onto the motor cortex and also the premotor cortex, and a lot of these are involved in motor learning. And the ones that you know the cerebellum's function the best is the vestibular cerebellum, and this is to basically to go to a synapse onto the vestibular nuclei from as what you remember from the previous slides, and this is a really important for balance and eye movement. So these are what a lot of people associate what a cerebellum to be with, and but actually when you kind of think about in terms of in terms of the area they actually is making up, this vestibular cerebellum is not taking that much room. Okay, now one of the things when it comes to the cerebellum is actually again a lot of it is involved in motor learning. Okay, and so one of the things that they did is to kind of make sure that what is the arm position and the associated EMG activity during rapid elbow movement. In A, what you're seeing here is that during a rapid elbow flexion in a normal subject, the agonist muscle produces like a burst to propel the limb. So this movement is braked by activation of the antagonist triceps. Okay, so basically, what you are seeing is that the agonist comes on and followed by a very fast opposing movement that is by the antagonist muscles. Okay, but what you're seeing in the cerebellar patient, which they have some sort of cerebellar damage, and what you're this fine timing of the agonist antagonist activity is really disrupted in patients with cerebellar lesions. And then so you can see that when the onset of the antagonist here, uh, over here is really delayed compared to the normal non-cerebellar patients, right? And then so that's one thing. And the second thing is that you can see there are a lot of ricochet movements in both the antagonist and the agonist arms. And so basically it's going to make that it's going to seem that there are some sort of oscillation that are at the endpoint movement. And so now this has to do this basically is a result from the attenuation of the agonist and the antagonist activity at the very first get-go, and then therefore the cerebral patient are not able to have a very swift movement, one jerk movement, but then just kind of stay in line. Okay, so this is one of the things that cerebellum does. It's really important to coordinate the agonist and the antagonist coordination kind of movement to make sure that you have a very smooth activities, muscle activities that's going on in your outer motor output. The cerebellum is also important when it comes to the motor learning, and this is the part I kind of want to talk about. And now, the thing that you can see that the lesions of the cerebellum really disrupt the learn response of something they call the eye blink conditioning. Okay, so what is the eye blink conditioning? Well, so you'll see that with the stimulus, basically kind of they want to pair a sound with the eye blink conditioning and the rabbit. So what you see here is that they'll have the tone that comes on first, and at the end of the tone, right before it's about to end, then they're going to give the rabbit an air puff right into the eye. Okay, and okay, okay. Now, what you see is that early on in training, the air puff causes the animal to blink, right? So the air puff is going to cause the animal to blink. So this is what you see in the beginning. Okay, so as and then, but what happens in the late condition training? You can see that the animals hears the tone and then anticipates that puff is coming, and therefore the blink is going to happen right before the puff comes in. Okay, so what this means is that the animal has adapted to this sound that comes on, and then kind of anticipates that, oh, there's going to be a eye, there's going to be a puff of air to my eyes, and then therefore I'm going to have to blink to kind of adapt for that response. Okay, now what happens to the animals in the cerebellar lesion? Well, the lesions in the deep cerebellar nuclei really abolish this learned response. Well, it doesn't abolish this response completely, but you see that the animals at the beginning phase is just like it's just like the unleashed animals. But in the late phase, you can see that they're not able to correctly time this very well, because as opposed to this time that's supposed to come on like around here, you can see that that the eye blink is kind of very premature, and in which that it was not able to have a correct timing of that. So the fact that the animal continues to blink reflexively in response to the air cuff kind of indicates that the lesion really produced a learning deficit and not a motor deficit. Okay, the anticipatory learned response are still present following a lesion of the cerebellar cortex, as you can see here, but however, they are timed inappropriately, and this it's no longer adaptive, right? Because it's not adapting to when the anticipated timing of that air puff is coming. Okay. Now let's take a look at the basal ganglia. Let's talk about it. Okay. So everything that you see in green is basically the basal ganglia. Okay. So you get the the VL thalamus, nucleus of the thalamus, and you got the caudate, you got the putamen, which makes up the striatum, you got the globus pallidus, and you got the subthalamic nucleus, and then you get the substantia nigra, specifically pars compacta, which produces dopamine. So these are all of the areas that are responsible, that makes up the basal ganglia. And so so all of anything that goes wrong within the basal ganglia could really can wreak havoc on your motor output and also your motor system, and as well as your motor learning. All of these things can really impair your ability to kind of move around in the world. Okay, now this is just basically kind of showing you where the basal ganglia are, okay? And so all of these structure we have kind of told you about already. So there is a caudate and putamen, which makes up the striatum. There's a globus pallidus, there's a subthalamic nucleus, and there's a substantia nigra, pars compacta specifically. Now, when it comes to the actual wiring of the basal ganglia, this is what the wiring diagram looks like. And you can see that all of these has an intimate connection to the cerebral cortex. So the white links kind of indicates the excitatory projections, and the black link indicates the inhibitory projections. You can see that the input from the cortex projects primarily to the striatum. Okay, and then from here, the processing flows along two pathways, and there is one that is the direct pathway, and there's another one that is the indirect pathway. Now, the direct pathway goes to the output nuclei, which is the internal segment of the globus pallidus, and then the pars reticularis of the substantia nigra. And then the indirect pathway includes a circuit through the external segment of the globus pallidus, and then to the subthalamic nucleus, and then to the output nuclei. And then now the output projection to the thalamus are relayed to the cortex, frequently terminating closer to the initial source of the input. And now the dopaminergic projections that comes from the pars compacta portion of the substantia nigra modulates the striatal activities by facilitating the direct pathway from the D1 receptors. So as you guys remember that the dopamine receptors, there are multiple types. So D1 receptors and D2 receptors, right? And then so D1 receptors are the excitatory type, and the D2 receptor are the inhibitory types. So the activities that is coming from the substantia nigra pars compacta really modulates the striatal activities by facilitating the direct pathway and also inhibiting the indirect pathway via via the D2 receptors. Okay, so so basically these are the activities I kind of wanted to talk about when it comes to this entire circuit that involves the substantia nigra. Now, when it comes to the two famous diseases that involves the basal ganglia, these are the Huntington's disease and there's the Parkinson's disease. In each one involves different neurochemical alterations. Okay, now what you're seeing here, the white link is again excitatory projections, and the blue link are the inhibitory projections. Now in Huntington's disease, the inhibitory projection along the indirect pathway from the striatum to the external segment of the globus pallidus is reduced. So the net consequences, is reduced inhibitory output from the globus pallidus, and thus an increased cortical excitation and movement. And then so this is why the Huntington's disease, it seems like they always have to move, and then they cannot control their movements very well. So this is the reason why, and because there's a lot of increased output, as you can see from the basically the reduction, the inhibition to the thalamus is really reduced, and then because of this reduction in the thalamus and from all of these increased inhibition to the globus pallidus, which cannot properly inhibit the thalamus, and this is why you see a lot of excitatory activities that's coming from the thalamus to the cortex, which really increases the number of the output that you see here from the cortex to the brainstem. Okay, you can see the arrow is much bigger. Okay, now Parkinson's disease is exactly pretty much like the opposite result, but the mechanism behind that is not the same. All right, so Parkinson's disease primarily reduces the inhibitory activities that's going on from along the direct pathway. So this is actually because the substantia nigra pars compacta activities that which not you're not seeing here is greatly being reduced in so basically what you're seeing that there's an increased inhibition that's coming from the globus pallidus to the thalamus, and therefore there is a reduction of the cortical activity of the movement, and which means that there isn't that much activity that's coming from the thalamus to the cortex, and therefore the kind of the output that from the cortex to the brainstem is also reduced. And this is why you see the Parkinson's patient really have a hard time moving their limbs, and their movement are slow, and then the postures are not really great. It's because all of these reduction that's coming as a result from the increased amount of the inhibition that's coming from the globus pallidus and substantia nigra reticulata, all of these areas are inhibiting the thalamus too much, and therefore the cortex cannot have the proper output to the brainstem and the spinal cord. Now let's take a look at the neuroimaging techniques that kind of let you know what is going on, what is going wrong in the Parkinsonian patients. Okay, so here what you're seeing is a PET study looking at the substantia nigra pars compacta when it comes to the dopaminergic activities. So you can see that in the healthy subjects, they have a lot of dopamine that is going on in the substantia nigra pars compacta, but not so much in the Parkinsonian patients. Okay, and then so this basically kind of this procedure provides an opportunity to visualize the reduced dopaminergic activity in patients with Parkinson's disease, and then you can see that the subject and the healthy subject and the Parkinson's disease patients, they were injected with the radioactive tracer, and then this and in case this is in case you're interested, there's something called a fluorodopa, and then so that this agent is visible in the striatum, kind of reflecting the dopaminergic projections to the structures from the substantia nigra pars compacta. So when you kind of compare the reduced uptake of the scan in the healthy subjects that you see in A versus the uptake in patient scan in B. So this basically kind of tells you that the Parkinsonian patients really have a reduction amount of the dopaminergic output that's coming out from this substantia nigra pars compacta. Now let's go back to our quick flexion task, okay? Now it's kind of similar to what you saw in the cerebellar patients. The Parkinson's patients also have a inappropriate response when it comes to these flexion. So what they're looking at is that the EMG data in the healthy people, the magnitude of the EMG activity really increases, produces, and it kind of corresponds to their the extent of their movement. So you can see that the big movement here also produces a big effect, and then the small movement produces a smaller effect, and followed by exactly the antagonist muscle movement. But when it comes to Parkinsonian patients, they have an issue with the bigger movement. Their big movement and small movement in terms of magnitudes are kind of similar. Okay, so you can see that what they are doing is more like a robotic kind of like this kind of movement. So this they kind of call this the cogwheel movement, and that's kind of similar to when you have a wheel that is not exactly spinning and that's perfectly round, but a cogwheel shape, and then you have this unsmooth movement. So you see this a lot in Parkinsonian patients, and then this is also reflected in their EMG data. You can see that in the big movement, what they do is they do not have a big magnitude of movement where you're supposed to see like this, but they have a many small amount of the movement. And then this is because the output that is coming from the motor cortex cannot adequately move these muscles enough, and therefore they have to kind of compensate that's coming from the thalamus to kind of keep going with this kind of excitation. So they have to give excitation in a short, small amount of time. And then so this is so therefore when you kind of think about it in the kind of neurons that the Parkinsonian patients really need to recruit on the motor cortex in order to produce one kind of movement is a lot more effortful when it comes to and getting something done. And so they're because they have to constantly recruit and increasing the recruitment of these neurons because their muscles cannot really kind of they cannot really accommodate this kind of task. But I want to kind of make sure that you guys know that this is not a muscle defect. This is a motor cortex defect because the motor cortex cannot give out the adequate amount of the data of the command basically, and since when that does not match with the actual motor planning that they have, that's either rooting from the SMA or from the PMA or coming from the dorsolateral prefrontal cortex, that they cannot properly recruit this amount of activity that's going on in the motor cortex. And then so then what happened is that the brain is going to have the feedback say, oh, that's not where it was, and that's not where I want to go, and I have to keep exerting this amount of the I have to keep exerting this amount of the of motor output in order to achieve my goal, essentially. Okay, so this is why you see this cogwheel type of movement in Parkinson's patients. Now, how can we correct Parkinson's disease? Well, so if this is a dopaminergic defect that in which that you don't have enough of it, then you can supply more dopamine, right? And as you remember from your behavioral neuroscience class, the precursor for dopamine is L-dopa. So what they do to what they give Parkinson's patients a lot in the beginning stages is they would supply them with L-dopa as part of those medicines. Okay, so that's the first method. The second method is deep brain stimulation, what you see in this graph. Okay, and then the deep stimulation is basically achieved by implantation of electrodes in the basal ganglia, most commonly in the subthalamic nucleus bilaterally, or the globus pallidus bilaterally. And then what it is a pacemaker device that is connected to the electrodes, and then it kind of implanted subcutaneously, and then the electrode can then be stimulated by the pacemaker at regular intervals to relieve any of the symptoms of the Parkinson's disease. So so basically the electrodes are the electrode's job is to stimulate the activity that's going on from the basal ganglion loop, so in which that can kind of correct this motor output deficiency that eventually that you see in the motor cortex, but that actually stems from this inadequate amount of activity that's going on in the substantia nigra pars compacta. Okay, now what other Parkinson's disease defects do they have? All right, so we're going to talk about two related tasks. Okay, so in this task, so there's a motor task and there's a cognitive task. Now in the motor task, the subjects that perform two successive sequences that were either identical or different. Okay, so they can have like the stimulus that is AA, but they can have the stimulus that is BA, and then basically they are asked like to play exactly the two sequence that are the same or two sequences that are different. And you can see that the control subjects, it really doesn't matter if there's no shift or there is a shift in the sequence, they do pretty well. But when it comes to Parkinson's, you can see that there is a significant difference when it comes to the no shift or the shift conditions. And for both conditions, when compared to the control and to the control patients, they are not doing very well. But they do specifically horribly on the shift stimulus. So which means that their motor planning activities is not that good. So they cannot have a sequence that are kind of not related and then kind of piece them together. Now, when it comes to the task, the subjects had to respond to either the color or the shape of the stimulus. And then so basically they have to do this in a shift or no shift. Okay, kind of similar to previous one, but then they're doing this in terms of different dimensions. Okay, and very similar to what you see before, the control subjects, they do pretty well in terms of like the reaction time. They don't take that much time, but the Parkinson's disease patients, they take a lot of time, and they do this when they have to shift between the dimensions. Okay, so what this means is that the both the Parkinson's in Parkinson's disease patients, in both the motor task as well as the cognitive task, they are especially slow when they have to do a mental shift. So which means that their motor planning is really defective, and that the way that they have to kind of shift their cognitive dimensions, the cognitive set shifting is also very defective. Okay. Now, so this is just basically the functional architecture of the motor system. Here, what this is just kind of sum up everything. So when you initiate with an idea, and then you have this, these ideas are being generated by the association cortex, and a lot of it is coming from, if you have an initial idea, that is coming from a very goal oriented, is coming from the dorsolateral prefrontal cortex, and if this is environmentally driven, this is coming from posterior parietal lobe. And either way, they're going to go to four places. They're going to go to basal ganglia, which is responsible for switching between actions, and they can go to the supplemental motor area, which is responsible for movement selections, reflecting the internal goals, set and the learned patterns, or they can go to the premotor cortex, which is really based on external stimulus information, or they can go to the lateral cerebellum, and what you can see, this is the preparation for the movement pattern. And all of these, eventually, they are going to synapse onto the motor cortex, and the motor cortex gives a movement. Now, of course, the motor cortex is going to have a specific feedback mechanisms that goes to places such as the intermediate cerebellum, which is responsible for the implementation and the correction of the movement patterns. But these are more on the lower level, and this is not on like the higher cognitive planning level that we see in these beginning areas. Okay, and then of course you get some sensory input that kind of lets the motor cortex know exactly how much you are exerting and what needs to be applied a little bit more or less, and so to speak. Okay, so this is going to